All right, well, I think we'll get started. Hello to everyone. My name is Tracey Steinrucken. I'm the president of the Australasian Mycological Society. Thank you for joining us today for our February virtual seminar. Before we begin, I'd like to acknowledge the traditional owners of the land on which we are all meeting today in Australia and New Zealand. I know we have some guests from Europe as well signing in, so thank you for joining us. I'm speaking to you today from Meanjin, Brisbane, and I recognise the country north and south of the Brisbane River as the home of the Turrbal and Yagara nations. I pay deep respects to all elders past, present and future. Our presenter today is Matt Salomon. Matt is a PhD candidate at the University of Adelaide. His thesis covers topics of urban agriculture and the application of arbuscular mycorrhizal fungi to improve soil health. Matt holds a bachelor's and a master's degree from the Technical University in Munich and has an extensive farming background. He is passionate about applied research and how existing technology can be used to improve sustainability in food production. If you'd like to ask a question or post a comment, you can do so by clicking on the Q&A button at the bottom of your screen. Matt will answer your questions at the end of his presentation. Alternatively, you can put your hand up, raise your hand at the end of his presentation, and you can ask your question via your video and microphone. At the conclusion of the discussion, please take a moment to answer the 3-question survey to help us improve the delivery of this series, as it is still quite new. So I think it's time to get started. Matt, go for it.
Thank you, Tracy, and welcome to my presentation and my work as a PhD student. As you can tell from the title of this talk, it pretty much gives away the results. At this stage, I just want to acknowledge that this study was not performed by me alone. I had, of course, my supervisors here in Adelaide, and actually this study, which I started independently, then turned into a global study, which also now includes research from Marcel van der Heijden at Agroscope in Zurich and Heike Bücking from the University of South Dakota. At this stage, I also would like to thank the Hans-Jürgen and Marianne Orff travel grant and the University of Adelaide for providing me with my PhD scholarship. And yeah, let's just start. I'm just trying to give a bit of a introduction into why we actually need mycorrhizal fungi before I come to the actual results. And just briefly, I guess you're all familiar with how the mycorrhizal symbiosis works. Just in case you don't, so we have here the plant root cells, and what you can see here are those structures called arbuscules. And these arbuscules are actually the organs for nutrient exchange, and you can see here the hyphae growing out from the root cells, producing spores, or you can see it the other way around, that the spores are germinating and growing into the root cells, forming those hyphaes. And the phylum of arbuscular mycorrhizal fungi is called Glomeromycota, and we have about 230 described species. However, most important commercially are just the Rhizophagus and Glomus, just because they are very ubiquitous and some can be seen as generalistic fungi. Arbuscular mycorrhizal fungi are also obligate symbionts, meaning that they need a host plant to grow and fulfill their life cycle. However, I linked a study there where just recently they used myristate as an alternative carbon and energy source, and they managed to grow and produce mycorrhizal spores in vitro. So maybe this for the future might be one thing that's be commonly more adapted. However, it's still research in progress, I would say. There are obligate symbionts for most of the terrestrial plants and pretty much all crop plants, and they can already be found naturally in most natural and agricultural ecosystems. Why are they important? They have a number of very good effects on plant growth, especially improved nutrient uptake, They provide heaps of ecosystem functions and improve soil health, carbon sequestration, water retention, nutrient retention, and so on. I just said that mycorrhizal fungi have a cosmopolitan distribution, and here's a study done by Fritz Oehl in Switzerland, and he looked at the number of spores that were naturally present in different cropping systems. We have here grassland, crop rotation, and monocropping, and we see that we have pretty high numbers in grassland, and it's strongly decreasing when we go to the monocropping. And just from my own research on urban agriculture, you can also see that we have similar numbers than in the crop rotation in the previous slides. So this is one of the sites that I'm visiting for my research. I collected soil samples, extracted the spores, and even there we have high numbers of spores, about between 3 and 5 spores per milliliter of soil, which might not sound like a lot initially, but if you think about the big surface area we have, it actually adds up to quite a lot of spores. And here's just a picture I found recently of what those spores look like under a microscope. These ones are about 80 micrometers in size. In case you want to improve your natural abundance of mycorrhizal fungi, there are major various practices you can follow. For example, instead of leaving the earth fallow, you can plant cover crops, or instead of plowing, you can use minimum disturbance like no-till or no inversion and so on. And this study here is done by Timothy Bowles and provides a very good overview of how to naturally improve the abundance of mycorrhizal fungi. However, this is not always possible. Just if you look at the cover crops, for example, here in Australia, most of the soils or most of the environments just don't have enough soil moisture to have more than one crop per season. Similarly, with the alternative tillage practice, there are just a few crops where you can't do it. For example, potatoes or beetroot. So it's not always possible. And in these cases, it might be economical to actually apply mycorrhizal fungi. And this here is a nice study where a commercial inoculant that apparently was working was added to various potato trials over 5 years. And we see that in 80% of all trials, the increase after inoculation was more than 0.67 tons per hectare in potato yield, thereby making the overall application of inoculum a economic situation. I might just add that in this situation with the potatoes, it is a bit easier because the seed weight per hectare is much, or the number of seeds, of potato seeds, is much less than if you look at wheat or barley. So it is a bit easier in these situations. Similarly, in this study, where they looked at Allium festusalum, and I think it's Welsh onion, and you can see here that they measure the shoot dry weight in accordance to different available soil phosphate levels. And we have control without inoculation and the inoculated control. And what's really exciting here is I think that the lowest soil phosphorus at about 200 parts per million, there are, with the inoculation, higher shoot dry weights than with the non-inoculated one at the highest superphosphate levels. And if you look at the costs of inoculating these seedlings, it's less than half than what it costs to apply so much superphosphate. Again, with the Welsh onion, it's a bit easy because you can just inoculate them in the greenhouse, and then once they reach a certain size, you just transplant them outside. So it also facilitates the inoculation a little bit. So we see it actually is an economic thing, and it makes sense to apply commercial inoculants in some situations. And so basically, that's also what I told my gardeners at the urban agriculture sites that I went to, and they asked me pretty much what's the best inoculant that I can buy, and I didn't know an answer, and I couldn't find much literature on it. So that's why we started to do an analysis of the microbial inoculants, and it's a fairly simple setup. We just bought bunch of commercial inoculants that were available at the time. We had a negative control, which is of course the non-inoculated treatment. We also added a positive control, which consisted of our self-propagated culture of rhizophagus, which is just there to see that our methods work, to see that, okay, if the inoculation works, if we add proper inoculum, then we get good root colonization. And then we added our various commercial inoculants. So in my study that I did here in Adelaide, I used tomato as a host plant. Marcel van der Heijden in Switzerland, he used leek. And then we both did the testing in sterilized soil. The idea there was to test whether there are any viable propagules at all, and then also to do it in non-sterilized soil, to see, okay, what's actually happening if we do it in a more applied scenario when there is a certain background abundance of mycorrhizal fungi. And here's just an overview of all the studies that's included in our work, which is going to be published soon. It's currently still under review, but hopefully published soon. So again, here the Australian and European studies, which are both just greenhouse studies in sterile and non-sterile soil. In my case, I always used a very sandy soil mix, so 90% sand. I always added the inoculum as recommended by the manufacturer. In the European study, Marcel decided to use a low P condition for the sterile environment and added 20 grams of each inoculum, which is of course a really big number if you add 20 grams to each pod. And that's really just the ultimate proof to see if there is any viable propagule in the inoculum. And then again, his more applied scenario in non-sterile soil, natural agriculture soil, which is then higher in P, and there he also applied the inoculum as recommended. Heike did a bit of a different approach. So that was a field study on soybean, and they used 3 mycorrhizal products which they had tested before and knew that they would actually work. And they counted the spores in the inoculants beforehand and then applied them in such a way that it's roughly 525 spores per square meter. And although this field study is a bit different scope than the greenhouse studies, we thought it's a nice addition to have in the paper. So that's why it's included, because it just shows you under the most realistic scenarios what would happen actually. And so I'm just going to start with the first results here. So you can see the mycorrhizal root colonization results in percentage. And you can see the Australian and European greenhouse studies using sterilized soil. And so what you can see first is that in both control treatments, we have zero colonization, which is as it should be because it's sterilized soil. There shouldn't be any viable propagules left. Then in the positive control of where we both use the Rhizophagus irregularis, with the tomato we get almost 50%, and the leek it's a bit more mycorrhizal responsive, we get 75%. So that also shows us that our system works theoretically, and it does lead to mycorrhizal root colonization if we add viable propagules. And then if we look at the Australian studies, so we have tested 10 different inoculants here, and none of the results are actually coming close to the positive control. with the highest one being here, treatment E and I with maybe 5%. So that's not really satisfying. And with the European study, we have 4 treatments that have the same colonization results and the positive control. However, we also have a bunch of treatments that obviously didn't work. And if you look closely, you can see that treatment number 10, 11, 15 are missing, and that's because no plants actually survived in this scenario. So after the addition of these inoculants, there's just 100% plant mortality, so no results in this case. I also want to present the results for the biomass. So the shoot and root dry weight added together. It's not really so important in this case because the mycorrhizal growth response is just very individual and can change depending on different ecosystems, soils, host plants, and so on. But still, just to give you a broad overview of what happened. And starting from the Australian study, we can see that treatment G performed very well, and that's because that one contained was labeled as a fertilizer, which also contains mycorrhizal fungi. So I guess it's not a big surprise that plants like fertilizer, hence the highest results there. Otherwise, there's not a clear trend between colonization and biomass. But if you look at the European study, there's a much better trend because, as I said, leek is very, very mycorrhizal responsive, and you have an almost linear increase in the biomass production with an increase in the root colonization. So basically all the treatments that resulted in high root colonization also resulted in the highest biomass production. And again, other way around, treatments with low root colonization also have lower biomass. Now switching to the non-sterilized soil or the field soil. Now it includes the North American study. But let's just start again with the Australian one. And we see now this time in the control treatment where we didn't add any further inoculum, we already have pretty high colonization, and it's just because on average with the soil that we used, there were about 1,200 spores present in each pot. So we already have pretty high colonization, and it doesn't increase after the addition of the positive control and of course any of the commercial inoculants. However, in treatment G, its colonization is completely diminished, probably due to the fertilizer content in this product. In the European study, it looks a bit the same. So we have quite a bit of colonization in the control. However, the rhizophagus positive control resulted in a big increase. And then the products that actually worked quite well in the sterilized soil, not working as well now in this case. And this could either be because we have now a high P condition in the European study, or because also in this study, Marcel has applied the inoculum as recommended and not this 20 gram for everyone. In the North American study with soybeans, so we again have a non-inoculated control, and we have a negative control, which is a fungicide treatment. So a systemic fungicide was applied weekly just to inhibit the growth of the mycorrhiza, and we can see first of all that the fungicide treatment worked. But then we see that there's not really any significant difference between the treatment and the control. So the introduction of a commercial inoculant into the field soil did not lead to increased root colonization. And again, I want to show you the biomass results, which are for the Australian study pretty much the same. So we see treatment G with fertilizer, plants respond very well to that. Now with the European study and leek, the results also changing and not as linear anymore, probably due to the higher peat content. And as you might see now, we actually have treatments included that weren't included in the sterilized soil. So it means plant mortality in the non-sterilized soil was lower when introducing those inoculants. And I think it makes sense because we just have a that's called a healthy soil microbiome, which, you know, after introducing certain unfavorable microorganisms could maybe counteract it again. So the plants actually survived. We have a bit of interesting results here, the North American study, namely fungicide treatment. Lower mycorrhizal root colonization did not lead into a reduction of biomass, but actually an increase. But also, so I didn't include the significant letters here, but treatment A has significantly higher biomass than the other treatments. And this shows that although mycorrhizal root colonization didn't change between the treatments, the introduction of a commercial inoculant actually changed the mycorrhizal community, and that way it favored the soybean growth, which resulted in a higher biomass production. I also want to show you this table. It shows the, so this was just done in the Australian study, I need to say, and what we did is did some nutrient analysis in the inoculants, and we have here our rhizophagus and our 10 commercial inoculants, and we tested mineral N, which is the sum of ammonium and nitrate, so the plant available nitrogen, and we have Colwell P, which is a common Australian measurement for the estimation of plant available P. And maybe let's start with treatment G, which is a fertilizer, and I guess as a fertilizer it's allowed to have a certain amount of nutrients, which it has, but then actually other inoculants have much higher nutrients than the actual fertilizer, and in this case the nutrients weren't labeled as such, and I think it's just a very strong case where gardeners or people in general would apply these inoculants and see a very good growth response, but actually that's just because of the nutrients in the product which weren't labeled as such. And also just briefly the spore count in the 3 studies. So we extracted spores and for all the products and counted them. And I included here the median. So the median in the Australian study was 18 spores per gram. European study a bit higher and much higher in the North American study, probably because they just used inoculants where they knew that they already would work. In the Australian study, there's quite a bit of range. So one product only had 1 spore per gram. However, another product had 715 spores per gram and still didn't lead to mycorrhizal root colonization, which just shows me that spores, although they were really present in a very concentrated form, didn't lead to, were just not viable anymore and didn't germinate. An European study was actually one product that didn't contain any spores at all. And North American study, also quite a big range in the spore concentration. When applied as recommended by the manufacturer, the median application rate was 34 spores per pot, which I think if you would apply 34 spores in close contact to a root and you have viable spores, it would be a pretty good thing. But I think if you just you know, think about it more in a broad scale scenario, that you can't put the inoculum into each little plant hole, you just don't have the time, you have to apply it in a more broader way. It's just not enough. European study, we have a bit higher median, and of course in the North American study, they always applied 525 spores per square meter. And here I have a picture of mycorrhizal spores from one product that did result in mycorrhizal root colonization. And we can see here the big thicker line is just a plant root, and attached to it a hyphae with the spores. And I think for me it looks like a rhizophagus species. And these bundles of hyphae, clusters, were found pretty much everywhere in the extractant. And if you looked at inoculants that didn't result in colonization, you had something like this, which were basically just, for me, it looks like dried and ground up roots. But you have to look very, very long until you find one spore. And this is the spore that I found in the extractant. I think we extracted 3 grams of, we used 3 grams of inoculum to extract the spores. So in 3 grams, I found this little spore here. So just to add a few further comments, issues that arise were the question about plant pathogens, which were not tested. There are various DNA-based approaches to test for plant pathogens, which we didn't do, but we had in some treatments quite a big plant mortality, which makes you wonder. A lot of the products missed a lot of information. Sometimes just said content, mycorrhizal product. We didn't know anything about the species or the concentration. Often they're used, advertised for use in hydroponic systems, which I think is a very doubtful assumption to make. As I said before, most of them were missing NPK analysis, although they contained a lot of nutrients. Some of them just had very unpleasant smells, which I think is not the best sign. Sometimes the carrier material was really hard to apply it, although we did it all by hand, so nothing was mechanized or anything, and even just applying it by hand, basically with a spoon, made it quite difficult in some cases. So what does this mean for the scientific community? I think whenever people are using commercial inoculants, they should definitely use a positive and a negative control treatment. For the consumer, I don't really know, because you can't test mycorrhizal inoculation with household items. I guess there's the people who are a bit more experienced there. There's the ink and vinegar method, but you still need a microscope and a minimum of lab equipment. I think there should be some consequences in terms of government regulations. For example, there is one recent approach with the new European Union fertilizer regulation, which now includes biostimulants and is specifically mentioning mycorrhizal fungi. And in March 21, so pretty soon, there's the deadline for the introduction of mandatory quality control methods, which need to be fulfilled by producers in order to sell the products. At least that's what I've heard so far. I also heard through the grapevine it might involve vitality staining of the spores. So not a plant growth bioassay is done in this study, but just staining of the spores to see if there's any metabolic activity happening, which of course can also be a bit misleading, because there's a difference between a spore being a theoretical viable and actually having enough viability to germinate in different medium and then actually colonize a host plant. We also that's the authors of the study that I just presented. We are following up with a opinion paper, which we call Establishing a Quality Framework for Commercial Inoculants Containing Arbuscular Mycorrhizal Fungi. We also invited a bunch of other experts in this field and producers. We already have a good dozen co-authors, and we just try to get a broad expertise, a lot of people into this opinion paper, so it has a high impact. And in this paper, we provide a example for a quality framework. In our case, a plant growth bioassay, which could be adopted by regulatory agencies. which then leads to the introduction of mandatory quality control methods as seen now in the European Union. Here's another picture of my urban agriculture sites, hopefully with lots of mycorrhizal spores, so they don't need commercial inoculants. By the looks of it, that's pretty much what it looks like. And I want to say thank you very much for your appearance today and for listening to my talk. Thank you.
Thank you, Matt. That was really enjoyable, and I think quite a bit of bombshell research for your PhD there that you've been doing, so congratulations. We don't currently have any questions on the Q&A panel, so if you're wanting to ask any questions, please go ahead and do that. You can also raise your hand at the bottom of the screen, and then I can activate your microphone and video. But in the meantime, Matt, first of all, what are the regulations in Australia like for commercial AMF inoculants? Are there any?
I don't think there are. I found regulations for composts, for example, and fertilizer, but not for microbial biostimulants.
Okay. So do you know if they are widely used?
I think they're not widely used. I know there's a trend in, for example, in the viticulture industry or horticulture that people use more alternative methods, such as compost teas and seaweed extracts to improve the microbial activity in the soil. I don't think that mycorrhizal fungi are used commercially at the moment, but I do hope that might change, and I think it might be a good option in cases of horticulture, all those almond orchards that we have, or viticulture, because you don't need a lot of inoculum to inoculate your whole field.
Do you think this is going to impact the organic farming industry in terms of if commercial organizations are not actually specifying what is in their inoculum? Is that going to have an effect when someone's actually trying to produce organic crops?
Yeah, I think just theoretically, if you apply any form of inoculant, which is basically just a potent mineral fertilizer, but not labeled as such, and you do it in an organic or biodynamic scenario, that's counterproductive. I don't think that it can be traced back, and I don't think the farmers are at fault at this stage because they're definitely misled. But I would think it could be an issue according to the principles of these farming methods.
Jeff Powell from HIE Western Sydney says, great talk, Matt. Were there any products that were tested by more than one of you, Marcel Heiker? And if so, did they behave similarly in each study, or were the products unique in each study?
So all the products were unique. So we have 28 different products which weren't repeated in the different studies. So for one thing, in Australia, we have very strict biosecurity laws, so you can't just import any mycorrhizal inoculant from other countries. So my products were just sourced from the Australian continent. And then I guess it was just luck that none of the other two, Marcel and Heike, actually used the same product.
Okay, and Pius Boris asks, or makes a comment. He just says that within the European framework, it now appears to be the case that mycorrhizal products are exempted for registration under the new biostimulants regulation. In this case, a quality control will not be in place.
Okay. That's a good comment to have. Maybe just go back to this slide. Okay, maybe just take it with a grain of salt what I just said. So for me, that's very new information which I just received from a German producer a couple of days ago, and I haven't had time to actually follow it up in depth.
Yeah, I guess with changes in EU structure and increasing regulations and everything, it's going to be a bit of an ongoing thing. I guess, the commercial companies to keep track of, but also it's important for growers and yeah, regular everyday gardeners to have to keep track of too.
Yeah.
Kirsten Ball says, thank you for your presentation. Do you think that the nutrients in the inoculants might have compromised the viability of the spores?
Yes, I do think so, especially in terms of the plant available Colwell P. I think it's not a big surprise that it's especially water-soluble P, which is by definition more plant available, has a detrimental effect on spore germination. And I think these concentrations of Colwell P are really extremely high.
And obviously there's a degree of commercial privacy in all of these, this kind of study. So you've obviously not included the names of the commercial entities at all. Have you had a lot of interest from companies who produce these products already in your paper?
I've came across some connections, mostly due to the opinion paper which we are now following up. But some other people have heard of my study, and somehow it made it to the producers, and they there seemed to be quite an interest from the producers, from what I can tell. But we are just very careful of how much information we do want to reveal, especially in terms of legal issues. And we don't want to point out any products, but just show the general trend, and show what sort of conclusions there should be made.
Yeah, excellent. And hopefully we can improve the regulation of products like these. I think that's all the questions. If anyone has a last minute one to pop up, you can go ahead. But thank you to you, Matt, for presenting. It was a really interesting talk. Good luck with the publications coming out. We look forward to reading them. For everybody else that was here today, thank you so much for attending.